Assalamualaikum and good evening. I'm the doctor. I am Nina Muhammad Sukri with matric number AW210104. For this individual assignment video, I will be explaining the usage of an analytical technique in the industry that I have been interested in, which is the cosmetics industry. The analytical technique that usually used in cosmetics is HPLC. What is HPLC? HPLC is an abbreviation for high performance liquid chromatography. It has also been referred to as high pressure LC. HPLC is a separation technique that involves the injection of a small volume of liquid sample into a tube packed with tiny particles, where individual components of the sample are moved down the packed tube column with the liquid mobile phase forced through the column by high pressure delivered by a pump. These components are separated from one another by the column packing that involve various chemical or physical interaction between their molecules and the packing particles. These separated components are detected at the exit of this column by a flow detector that measures their amount. And output from these detectors is called a liquid chromatogram. In principle, liquid chromatography and high-pressure liquid chromatography work the same way, except the speed, efficiency, sensitivity, and ease of operation of HPLC is vastly superior. And what is HPLC used for? The first one is separation and analysis of non-volatile or thermally unstable compounds. The next is the identification of individual compounds in the samples, the measurement of the amount of a compound in the sample or concentration. The fourth one is the preparation of pure compound, and the last one is trace analysis. Preservatives are added to personal care products at relatively low levels to ensure products remain safe and perform as intended over their lifetime. The determination of preservatives in this product is important for quality control to prevent allergic reaction and other health issues. Preservatives are added to personal care products at relatively low levels to ensure products remain safe and perform as intended over their lifetime. The determination of preservatives in this product is important for quality control to prevent allergic reaction and other health issues. The most widely used preservatives in cosmetic products are a class of compounds generally referred to as parabens. These compounds are alkyl ester of p-hydroxybenzoic acid. They are used for their preservative properties in cosmetic and personal care products because of their antimicrobial activities, low toxicity, and low production cost. Methyl paraben is found in nearly all cosmetics. The use of parabens in personal care products has caused concern because of their potential adverse effects, including proliferation of breast cancer and reduction of sperm count and testosterone level. The FDA finds that although parabens can mimic estrogen, the levels found in these products are at such low levels that their activity of the body does not cause cancer in any higher incidence than naturally occurring estrogen, despite contrary belief. However, when counterfeit products make it to market, they are unregulated and may contain preservative levels that may pose a health risk to the user. Therefore, accurate methods to determine the levels of these compounds need to be available for monitoring preservative concentration in cosmetic and personal care products. The most frequently used technique in preservative analysis is high-performance liquid chromatography (HPLC). A reverse phases is typically used with gradient elution by a mixture of water and organic solvent. Gas chromatography and capillary electrophoresis have been reported much less frequently, and single determination have been made use spectrophotometric or photometric methods. Chromatography is so widespread, it can really be found in any field for any applications. But in case you are wondering if we really use the technique in cosmetic, the answer is a clear pull yes. But I suppose you will like more than a skin-deep explanation about how to use flash chromatography and preparative HPLC to produce sunscreen and eyeshadow. So here it goes. During cosmetics production, chromatography systems are typically used in the initial part of the process during ingredient discovery and purification. Active constituents used in cosmetics can either be synthesized in a laboratory or extracted from crude natural products. Natural product extracts often consist of hundreds of compounds, and you often need to isolate a single compound of interest from the mixture. Most commonly, we perform the initial isolation by flash chromatography to pre-fractionate and reduce the number of components in the mixture. Then we follow up with a final purification by prep HPLC. Flash chromatography is suitable for the purification of high quantities of both synthetic and natural products. It is an ideal technique for rapid fractionation of crude extract or coarsely purified fraction. When we use flash chromatography, our main goal is to reveal the most active fraction in the mixture so that we can correlate their bioactivities to the molecules that they are made of. For isolation of active ingredient, the sample can be introduced either by liquid or solid loading to a cartridge. Typically, we use normal phase or reverse phase silica with a particle size range of 15 to 50 micrometer as a stationary phase. Often, we need to collect several fractions for further characterization, bioactivity testing, and further purification by HPLC. Speaking of preparative HPLC, this method is usually used in the final step of bioactive material purification. For the technique, we most often inject the sample via liquid loading on a steel column with silica as the stationary phase. Preparative HPLC is a fast, flexible, and robust method of separating compounds from complex mixtures at high purity. The technique has a better resolution than flash chromatography because of the smaller particle size of the silica, among other reasons. The smaller silica size of the stationary phase, around 5 to 15 micrometer, result in a bigger surface which the solid and sample can interact. This improved interaction between mobile and stationary phase increases the resolution. Another point I want to bring up is the type of stationary phase and mobile phase or solvent that you choose when making cosmetic products. As expected, the materials selected for isolation of natural product by chromatography depends on the type of compounds present in the extract and the extraction process used prior to the purification. For example, the extract of a plant obtained with aqueous ethanol will be substantially different than compounds extracted with hexane. Certainly, polarity of the compound mixture is a major factor in selecting the type of mobile or stationary phase in your purification. Previous methods have determined single preservatives or preservative in a mixture with drugs or other preservatives, but never the simultaneous determination of the five mentioned preservatives. Moreover, the described method of sample preparation of chromatographic analysis have only focused on specific product categories. There is currently no universal approach to this type of analysis, either in developed chromatographic methods or sample preparation procedures. I think that's all from me. Thank you for watching.